This is Winchester Academy.
Thank you. Thank you. Hopefully I'll have time to actually give the talk. Thank you also to the organizers for allowing me to come back. Raise of hands for those that have seen or heard me speak last time. Okay, then half of you guys can probably leave because it's going to be exactly the same thing. I'll try to make it a little bit different. But I'll start off very much like before and give you a very basic introduction about genetics. So everyone in this room has equal footing, and hopefully you'll understand a lot of the content that I'll be describing. So I'll start off with a Genetics 101. I'm going to follow up and talk about some of the resources that we have at Marshfield Clinic, and specifically how we use electronic medical record to do a lot of the research that I'll be describing. And then the last part will really be about the research. And I'll talk about 2 different studies, all leveraging the electronic health record. And there's questions that you can ask yourself. These are the questions I ask myself when I'm doing the research is, what can we learn from families? What can we learn from populations, a whole group of people? Maybe there's populations of families within this room. And obviously everyone has a family. But then the big question is, how can we use this information to hopefully advance healthcare? And these 2 projects that I'll be describing includes a very large twin study, which will be actually a continuation of what I ended with at the last time, where I was giving sort of theoretically what we were going to do. And actually I'll be able to show you some of the research that we actually came from that twin study. And then I'll talk about another study, again, it's all rooted in genetics, and that's called Pharmacogenetics, which is a specialty of genetics, which is trying to understand how we, based on our genetics, can respond to drugs. So we got this anonymous gentleman. Again, this is how I started it last time. But like this person, as well as everyone else in this room, we're all a composite of millions of cells. And inside those cells, we have a very special compartment. It's called the nucleus. And with my trusty pointer here, the red glob, that's the nucleus. And it can be different colors. I'll show you in a couple slides nucleus that are blue. But nonetheless, inside those nucleus, we have chromosomes. And we inherit 23 chromosomes from our mother, and we inherit 23 chromosomes on our father. Now, we have a very special part on our chromosome at the very end. They're called telomeres. You can envision them as protective caps. And when we're born, we all are born with long telomeres. Now I'll describe a little bit more about DNA in the next slide, but these are long stretches of DNA that protect our cells, and we have special machinery in our body that actually adds these caps. And the mechanism is called telomerase, and there's a very special gene called TERT, telomerase reverse transcriptase. Don't worry about the names. If you can't remember TERT, think of dirt. It's highly repetitive, and as we get older, these telomeres get shorter and shorter. And if we get sick, our telomeres can get shorter and shorter. And we actually have an internal biological clock. So when the telomeres get so short, the cell will start to die. So and it's actually hijacked by cancer, because cells are always dividing, and they don't get any older, actually, because they can modify this mechanisms. And I'll talk about that and how that's related to the research in a little while. But I wanted to introduce you to this in the beginning. And as I mentioned before, you can ways of visualizing a nucleus. Here's another one. This one, in case, is blue. You don't see the outside of the cells. It's just a staining of the nucleus, and there's a whole bunch of them, a whole bunch of cells. And each one of these might be hard to see from the back, but if you look, the green, at least they're supposed to be green, little dots, is a way of visualizing these special telomeres. So those are the protective caps that we have on chromosome, but you have a lot of DNA that's in between both ends of your chromosome. And these are long polymers. It's a long chain of what's called nucleotides. We're more familiar with A's, C's, G's, and T's. It's the code of life. And what makes me different from everyone else in this room is that we have genetic variants within our body. You might be more familiar with the term mutants, but we typically don't use that term because it has negative connotations. It has inferences in function and everything else. But one person in this room might have a very, very specific position, a T, within their genome, whereas another person might have a C. Again, this is what a genetic variant. And these variants can have very large or small effects on who we are. And they can include health. So I'm giving a hypothetical gene. So I should back up. So these nucleotides, and there's functional parts of our genome that we call genes. So these actually create our proteins and everything else. And sometimes you can have a particular variant in your gene, in this case our health gene, that maybe increases your risk for multiple sclerosis. Maybe you can inherit a different variant in the same gene that might increase your risk for diabetes. Maybe you can even inherit a variant that's outside the gene, but maybe it affects the function of the gene, increases its expression or whatever. And maybe it doesn't actually increase your risk. Maybe it's actually protective. In this case, maybe it's protective against infections. But more often than not, what we have are variants that could be inside the gene. They could be outside the gene. They're called benign variants. For as good as we know, they may not have any function whatsoever. And what keeps me employed is that it's my job to identify the few biologically and maybe clinically relevant variants in a sea of millions and millions of benign, non-functional variants that all of us inherit. So I'm happy to, if anyone has any basic questions about what I described. Otherwise, if there's more pressing conversations, we can save it to the end. But I want to make sure everyone understood that basic concept, telomeres, genetic variants. OK, so now we'll move on to Marshfield Clinic and some of the resources that we have to use to do our research. So Marshfield Clinic, we've been around for now over 100 years. We just had our 100-year anniversary last year. We have approximately 60 sites across central, northern Wisconsin, even into upper Michigan. We have 4 hospitals and growing. We actually just acquired St. Joseph's Hospital within Marshfield, so we've captured all the healthcare within Marshfield specifically. We have multiple dental clinics. We have our own insurance company. Maybe some of you are clients of Security Health. Marshfield Clinic is one of the largest healthcare institutions in Wisconsin. We see about 4,000 patient encounters, which represents approximately 300,000 unique patients every year. And as you can appreciate, many of those patients are in rural communities. Many of them come from agriculture populations, families that grow up on farms. So our populations don't move in and out of the communities very often. We have a very stable patient population, which is fantastic for a lot of the research, in part because we have also an extensive electronic health record, or electronic medical record system, which started in 1984 as one of the first to be built within the United States. And we have data actually within that system that goes back to the 1960s, with over 2.6 million patients within current and past patients within our system. Also, which will be important with some of the research that I'll be describing today, is we have our own biobank of about 20,000 patients who volunteered to allow us to have their DNA as well as look at their electronic health record data. Now, within the electronic medical record, there's a wealth of information. You can have doctor's notes that are in there, so we have text data, we have prescription records, we have heights and weights and other biometrics, but we also have diagnostic codes, specifically ICD codes. These are actually codes that were maintained and developed by the World Health Organization, and it's a hierarchical coding system to define disease when patients are being treated. So if you go into the hospital and complain that you have chest pain and you get a large workup, you might, and maybe you're actually diagnosed with a heart attack, there'll be a very specific code that defines the heart attack. And it might be very specific codes that define the type of heart attack someone might have. Now, the challenge with these codes is that you go to the hospital and you have chest pain, and the hospital does a very large workup to see if you had a heart attack. Well, the hospital might still code you as having a heart attack because these codes in the United States are primarily used for billing, and they want to be paid to treat you for as having a heart attack, even if you hadn't. But nonetheless, they do work pretty well in defining people that have and don't have a disease. And they allow us to look at diseases at different levels of resolution. And I'm giving you examples where we could take a very specific code, 414.1, which defines aneurysms of the coronary vessels. And it can go to different levels. I'll start down here. So we have a very specific code, 414.11. And all these like codes can be binned into 414.1. All these like codes can be binned into 414. And again, it just goes up. And what I'm describing here is sort of also reiterated above, and just to help you understand some of this coding system. And again, it's a hierarchical system. So we know who's diagnosed with breast cancer, 174.9. We know who has diabetes. It's got a code 250. Even if you go in because you have an ingrown nail, 703.0. But it can get kind of crazy. There is a code for injury due to yoga. My sister is a big fan of yoga. Maybe she's had this. I don't know. Now, there's some other codes that are quite unusual. Maybe codes that might have been relevant 100 years ago. So if your animal got an injury on a streetcar with an animal-drawn vehicle, there's a code for that. And maybe the bright people planning for a future, there's a code actually for getting injured on a spacecraft. I don't know how often this happens. Incidentally, about October of 2015, we actually transferred to a new coding system. So we were ICD-9, and we have enormous amount of ICD-9 data, and this is really what we work with because we have so much longitudinal data with ICD-9. But ICD-9 was recently replaced, again in 2015, with ICD-10. Again, there's still another hierarchical coding system, but it adds additional information. And like the ICD-9 code, there's some funny codes in there. So one code is getting struck by a duck. I don't know how often that happens in the United States, but again, this is an internationally recognized system, so maybe there's other parts in the world where getting struck by a duck happens. There's a code for getting sucked into a jet engine. And now, that would be tragic that if you get there, but if you survived, there's a code for getting sucked into a jet engine again. And I think one of the strangest codes is that there's a code for getting burnt due to water skis on fire. Needless to say, I'm not studying these diseases or these conditions. But we try to take advantage of all the health information within the medical record, specifically these ICD codes, to study thousands of diseases simultaneously, which includes studying breast cancer, which includes studying diabetes, as well as getting struck by a duck. Okay, so now more to the interesting part, more to the research. And the first thing I'm going to be talking about is my twin study, but I'll take a step back and really emphasize the importance of families. And so here's obviously a family that's getting married, and then I imagine by looking at this picture you can have appreciation of who belongs to which family. So height is highly heritable. So there was a famous author that once said, "Family faces are magic mirrors. Looking at people who belong to us, we see the past, present, and future." Now, I don't know how many people were ever warned before you got married to look at your spouse's parent of same sex. And I was just glad that my wife still decided to marry me after seeing my father. But to further emphasize on who we are is a lot about what our parents are. And we have my 3-year-old daughter, who is now 10, and then we have my wife, who is a little frumpy there, and the picture's not as clear, but there's a lot of similarities of who we are based on where we come from. And so a couple of years ago, we did an extensive family study where we wanted to identify relationships within our electronic health record. I mentioned that we have 2.6 million patients in our system. And like many family members that presumably at one point lived together and they have the same last name, same home address, contact information, billing information. So we took advantage of that information. We de-identified it so that all Smiths have some random number. So I don't actually know who is who in the system, but I know that these people have the same last name because they have some random number that's associated with it. And all Johnsons have a different number, so we can relate people with this de-identified information. And on the left side is just kind of showing a decision tree on how we just define these relationships. But in the 2.6 million patients, we were able to identify about 27% of all those individuals that were related to someone else in our system. So in other words, 700,000 Marshfield Clinic patients could be linked to approximately 170 or 190,000 families or pedigrees. And this is a reflection of all that family information. I don't know how often you've seen these pedigrees, but circles represent females, males are squares. These diamonds, they're unisex. We don't really care in this case of graphing whether or not they are males or females. But what we did, and to sort of explain this figure, is take a family, a pedigree, and take the youngest person in that family, and you define them as your anchor. And then you do that for 190,000 families, and you stack them all on top of each other, and now we're looking down at them. So every family has that anchor, 100% of the families. 45% of the families have a second child or more. 81% of the families, we were able to identify the mother, 63% of the father, and these pedigrees go out and up. So the largest family we identified is over 130 individuals. And we even identified a family with 5 generations within our system, which reflects the amount of longitudinal data, the amount of time data that we have within our medical system, because our patients, again, don't move in and out. But a unique family relationship that we can focus on within these families includes twins. So looking at all this family information, we were able to identify approximately 16,000 twins, or about 8,000 pairs of twin families. And twins are very unique, in part because some of those twins represent identical twins. The scientific term is called monozygotic. And they're unique because they're one of the few family relationships where two individuals share nearly 99.9999999% of their genetic makeup. So they're, for all intents and purposes, they are identical at the genetic level. And I'm giving examples of 3 pairs of twins that many of us might be familiar in pop culture. On the far left, we got the Weasley brothers from Harry Potter, something that I grew up with seeing on TV, Mary-Kate and Ashley Olsen. Some twins more recently became popular, includes Scott and Mark Kelly, in part because both of them are astronauts. One of them was married to a senator. And if you're interested in twin studies, they just recently published a paper. It was really cool in trying to understand the relationship between what space can do to a person. And so one of those twins was actually in space for over a year. And which is kind of cool with that study is the person goes up, and because of Einstein's theory of relativity, when they came back, they're actually different in ages than what they were before the study began. Now, those are identical twins. The other type of twin is fraternal twins, or dizygotic twins. Now, they share 50%, in general, 50% of their genetic makeup. They're actually no different than any other sibling relationship, other than they share the same birth date. Now, here's some less famous twins. This is me, and this is my sister, who's sitting in the front row. Now, you can obviously tell we're not identical, and it's impossible to be identical twins if you're opposite sex. But with all twins, whether or not you're monozygotic or dizygotic, what makes these powerful families to study is because they typically share the same environmental exposure, especially early in life. But by studying twins, looking at monozygotic twins and dizygotic twins, you can begin to parse out the genetics versus the environmental influences in human health. So we did a study focusing on twins with the hypothesis. The question that we're asking is, can we use electronic medical record linked twins to study thousands of diseases, which we call phenome-wide research, to identify diseases that may have genetic influences, genetic etiologies. And the methods for this is we focused on our 16,000 twins at Marshfield Clinic. Incidentally, using our same methods, we identified approximately 12,000 twins at Mayo Clinic. And then we studied all the diseases, looking at those ICD codes I described before, over 5,000 diseases, which includes diabetes, breast cancer, hanging or ingrown toenails, and anything else. And what's a challenge with these twins is we actually don't know zygosity, but because they're related, we can just sort of ask a very simple question. What is the risk of one twin being diagnosed with a disease if the other one's been diagnosed? So that's called measuring a recurrence risk ratio, and we do that across the whole population of twins. And what we saw was very striking. Now, a reoccurrence risk ratio of 1 means there's no increased risk. A reoccurrence risk ratio, something greater than 1, means that you have an increased risk, and a reoccurrence risk ratio less than 1 means that you have less risk. It's protective if your other twin's affected. So under a random model, you would expect most diseases to hover around a value of 1. That means that there's no increased risk or decreased risk. It's just totally random. Now, it never is really at 1. It's just if you look at 1,000 diseases or 1,000 tests, is that you'll see this curve around 1. So imagine flipping the coin 10 times, and you get heads 5 times, and you get tails 5 times. 5 over 5 is 1. You flip it again. Maybe this time you get 6 heads, 4 tails. So now it's something greater than 1 if you take that fraction of heads over tails. You flip it another 10 times. Maybe this time you get heads only 4 times, and you get tails 6 times. So now it's 4 over 6, something less than 1. And then you flip the coin 1,000 times, or these 1,000 cycles of 10 times. You might have an instance where you get 9 heads and 1 tails, or you might get another instance of 1 heads and 9 tails, just by chance. And that's the same thing that with a lot of the statistics that we do. And when we look at thousands of diseases, we can evaluate whether or not what we observe is by chance. And what we observe within our twins, whether it's Mayo or Marshfield, is 99% of the diseases. I know this is just all blue, but trust me, there's a little sliver here. 99% of the diseases have these reoccurrence risk ratios greater than 1, with 1% having less than 1. Again, the random model should be somewhere hovering around 1. You see this even distribution. So under the random model, you see 50-50 if I was graphing it out here. And what we observe with Marshfield data is the same thing with Mayo data, with these twins, which just reiterates that families are critically important in our health, in part because we're sharing the same environment, but we're also sharing the genetics. Now, when we evaluate the relationships between these risk ratios, and I know that the scales are a little bit unusual, so try not to focus on the scales, just look at just the broad picture. But what we're showing here is when we look at the diseases that are shared between Marshfield and Mayo data is that there's high correlation. So those diseases that have really large reoccurrence risk ratios in Marshfield, that's on this scale. If we follow that, what we see is typically most of the Mayo data here also has, for that same disease, a large reoccurrence risk ratio. Ones that are low on Marshfield, they're typically low on the Mayo data. So again, iterating that we have some reproducibility in the results that we're observing. But of course, I'm a geneticist, so I'm trying to understand what is driving these conditions. I'm studying thousands of diseases, but I want to focus on, well, what are the genes that are influencing these observations? So we actually took these reoccurrence risk ratios and we binned them into groups. For example, we focused on these diseases that have reoccurrence risk ratios less than 1. That's actually this little tiny sliver on the top that I'm trying to show in there. That's its own category. And then we take diseases that have recurrence risk ratios that range between 1 and 2, 3 and 4, 4 and 5, et cetera. And then we got really strong ones. These are ones that are greater than 1,000. Those are the ones way over here. And then we asked, what % of those diseases are known genetic conditions? Cystic fibrosis, obesity is one of those. There's a number of diseases that we know that we can take from the ICD coding that we can define as genetics. And what I hope to show you, both within Marshfield and Mayo data, is that this is the proportion of genetic diseases here. As we increase in these recurrence risk ratios, as we get into larger and larger recurrence risk ratios, meaning that if one twin's affected, you have a higher probability of also being the disease. As we get to those conditions, what we see is an enrichment for known genetic disorders. So when we focus on these, the ones that would be perceived as protective, recurrence risk ratios less than 1, we see somewhere between 7 and 8 of those conditions being known genetic conditions. Whereas when we look at those extremes on the other side, about 21% of those are known genetic diseases. So we have a threefold enrichment for genetic conditions. That means, in this case, 20% of them are known genetics. I'm not interested in studying known genetics because they're known. I'm trying to understand the 80% other that haven't been described. They might still be environmentally influenced, but again, I'm trying to understand the genetics. So we take advantage of all this information as well as other stuff that, unfortunately, I don't have the opportunity in time to describe. But we took all this information and tried to prioritize which diseases were the most interesting that could be genetic. And what we focused on is a condition called seborrheic keratosis. It took me 6 months to learn how to say this. But if you say it enough, you eventually learn it. But really, it's a benign condition. And I would imagine many in this room might have been told by a dermatologist that they have a lesion called seborrheic keratosis. It's very common with age. In this case, it's actually, when it covers large part of the bodies, it could be a symptom of cancer. So there's a relationship between cancer and seborrheic keratosis. But for the vast instances that many of us will get, they're just random events that are occurring on our skin. It has nothing to do with cancer, at least not what we think. And so we went back to our biobank, and we, using the ICD coding, and identified, well, who has a code for seborrheic keratosis. That represented about 3,000 individuals. And we identified who didn't have it. That represented about 10,000 individuals where we have genomic information. And we did what's called a GWAS. And I sort of described this last time I came here. In a GWAS, what you're doing is you're looking at millions of variants, and you look at each one and you just ask, is the prevalence of that variant more common in your case group, the people with the disease, versus the people without the disease? And then you can calculate a statistic, and you get this called p-value and everything else. And that's what I'm graphing you here. This is 8 million genetic variants from our GWAS. And the larger these data points, so each one of these data points represents an individual genetic variant. The higher it is up on this graph, the more interesting it becomes. And anything that becomes over this line, we're excited. We throw parties, have cake, because that doesn't happen all the time because of that. Like I mentioned, you flip a coin a million times. At some point, you might see something interesting. This is a very strong p-value, but it's not interesting because we flipped the coin 8 million times. So we look for signals that would go past that noise. And what we found is multiple variants in a gene called TERT. I don't know if you remember from the beginning, but this is telomerase reverse transcriptase. It rhymes with dirt, and it's important in our telomeres. So we went to our tissue bank and looked to find people that had seborrheic keratosis removed from their skin. Often people might have these removed because they don't like them on their face or whatever. A clinician might be worried it could be melanoma or something. So they might be removed just to rule out that they might have melanoma. But we have all these tissues that are available for research. And so we took this tissue, and then we wanted to measure telomeres. So I showed you that slide before. Again, we have our nucleus, and each one of these dots represents the signal that we get from telomeres. And you can, let's say we'll take normal here. So on the right side is normal. As you can see, we got a very meaty lesion here, seborrheic keratosis. Anyone want to guess what the next experiment is? Yell it out. We'll look at the seborrheic keratosis tissue. So we just looked at this. That's here. And now we have seborrheic keratosis. Again, we have these blue nuclei. But I hope you can appreciate, I realize it's a little bit harder to see on this slide, but there are fewer dots. The intensities are not nearly as bright, which means that the telomeres are missing, or they're really short. And so we actually measured this in 20 samples. And what we showed is, of the 20, 16 of the samples had loss of telomeres. So we flip a coin 20 times, and you get 16 heads in a row. That's a weighted coin. This is what we observe. It's statistically significant. And these results suggest that, based on our GWAS, based on our genetics, that an important gene that's involved in maintaining our telomeres, which is critically important for our health, is also lost in people that have the lesion for seborrheic keratosis, for the very specific part in the skin. And so I really like this story because it starts with the big picture. We're looking at thousands of diseases, phenome-wide. Then we focus in on one disease that we think might be genetic, and we identify multiple variants within a specific gene involved in telomere maintenance that predisposes people for seborrheic keratosis. And then we further go on to the biology to define that telomeres are important for the pathology, for the etiology of seborrheic keratosis. Now, unfortunately, this isn't necessarily a disease I can put a career on because NIH or whoever doesn't care about these benign lesions, so getting money to study this extensively is difficult. But nonetheless, it's interesting because the gene itself has such an important role in cancer. So by studying people that have these lesions, which are relatively easy to acquire, most people that have them don't want them, so they're more than happy to donate them. By studying them, we can gain some insight into cancer because of the importance of telomeres in cancer. Now, what's really cool about this, and I don't know if I really have a lot of time, but these variants that are associated with telomere or with increased risk for seborrheic keratosis actually protects people from breast cancer. So there's obviously some sort of connection that's worth studying further. Now, again, I'm studying thousands of diseases, focusing on one specific one here in the end. But again, every disease or any phenotype that we focus on is captured in these twins, which includes drug response. And that comes to the second study, which I talked about pharmacogenomics. I sort of call it the study of the dead. Because in this study that I'm describing, and it's a very different study, so I hope you can tolerate the transition that I'm moving towards. But what we're trying to understand is by studying dead people, where we have their genetic information and we have lifetime medical record data, we can ask the question, well, what diseases or what treatments or how could we have treated these patients any different had we known their genetics? So we can ask this retrospectively. Now, specifically, we're focusing on can we prevent adverse drug events. There's very specific codes for adverse drug events, so we can extract some of this information. And so the hypothesis we're asking is, knowing an individual's genetic profile, can we use that information to determine who would have been at risk for an adverse drug event? Now, adverse drug events are extremely common, ADEs, about 125,000 hospital admissions every year. If you have it in the hospital, it usually results in an extended stay between 2 and 5 days, over 3.5 million visits with the physician as a result of these. It can be deadly, over 106,000 deaths every year. And what's interesting is that the vast majority of the adverse drug events are preventable. And this is in part because there's a lot of genetic information that contributes to this. So we know, scientists know, the FDA knows, some physicians don't necessarily accept this, but the governing bodies know that there are over 60 genes that's involved in the response to over 190 drugs that many of us are taking. And you can actually simplify this further, that only 5 genes can explain the response to about 42 drugs, and many of those drugs are some of the most common drugs. For example, codeine. So codeine is called a prodrug. This is a drug that, when it's a prodrug, the drug you actually take is not therapeutic. Your body actually has to convert it to something else to actually have it work. So codeine is metabolized by a special enzyme called CYP2D6. Again, the name of the gene is not important, but it's converted to morphine. And morphine is the part that has the analgesic properties. It's the therapeutic properties. And the Food and Drug Administration recognizes that there are genetic variants that many of us carry that will affect how we metabolize codeine. And as such, there's a black box warning on codeine to indicate to physicians or pharmacists that patients should be potentially tested genetically first before they're given this drug. And I'm going to give an example of one such case that has happened previously. So we have a new mom. She's nursing. Unfortunately, she had she had complications during delivery. She has enormous amount of pain postpartum. The doctor prescribes her codeine. After 7 days, unfortunately, the baby dies in its sleep. They attribute it to SIDS, sudden infant death. So very tragic. 7 or 10 days after birth, the mother starts having bradycardia, slow heart rate, and she's having other symptoms. She's rushed to the ER. They do this blood workup. They find that her levels of morphine, blood morphine, is 10 times higher than what would be therapeutic, the therapeutic index. So she's got too much morphine in her blood. They give her a genetic test. She's identified as being an ultra-rapid metabolizer. So she has a very specific variant. That means that all the codeine that she's taking is quickly being converted to morphine. And so over time, that morphine is building up and building up. And unfortunately, that morphine got into her breast milk, killed her child, and now she's having additional adverse drug events, all because of her genetics. So to understand, based on genetics, can we use this information to predict whether or not, or to ask after the fact, after these patients are all dead, not the example I gave here, but could we have done something differently in the context of pharmacogenetics? So we studied 300 patients in our biobank who are deceased. We have lifetime medical record. They were born in Marshfield Clinic. They died in Marshfield Clinic. And we looked at all their genetic information. We sequenced their entire genomes, focused on these 60 genes that I described just a few minutes ago, looked at their variants, looked at all the drugs that they were taken over their lifetime, and asked to see, well, what happened? Or maybe even ask, well, what is the potential of pharmacogenetics? Like, who could or couldn't or should or shouldn't have had genetic testing based on their entire lifelong medical records? So this is a figure. I recognize it's going to be a little bit busy, so I'll try to walk you through it. So let's focus first on the red. Here's our genes, CYP2D6. So we sequenced these 300 individuals. What we found is actually 9 genes total. Of the 60 genes, only 9 of them we identified variants. And under 300, about a little over 30% of the individuals in the 300 carried some variant in their CYP2D6 gene. Whereas we have another gene right here, VCOR1. So people that inherit this variant, they don't metabolize or they don't respond well to warfarin, very common drug. About 50% of the population carried a variant. And when we look at all the genes combined, over 90% of the 300 people carried a variant. Now, it only means something if they were given the drug. So 9 out of 10 people in this room are carrying the variants, but these variants only matter if they're carrying the drug. So we can go back to our electronic health record data and see who was given what drug. And that's what these blue bars are representing. So we'll go back to CYP2D6. So I mentioned here, about 30% of the patients carried the variant. But based on the drug data, just under 30% of the people not only had the variant, but were given the drug. And in fact, they were given one of 26 different drugs that may be impacted by this particular gene. Now we look at VCOR1. I mentioned about 60% or so. When we look at people that actually were given the drug, there's only one drug, warfarin. About 30% of the population had the variant and had the drug. And when we go all the way back to this side, I talked about 90% or more actually had the variant, but about 80% of the population not only had a variant, but also was given a drug that could have been impacted by the genetics. So that means almost everyone in this room will at some time in their life be given a drug that could be impacted by some variant inside them. Now that doesn't mean that they're all going to have adverse drug events, but it just means that there could be higher probabilities. And one of the things we also wanted to look at is, well, what is on average the number of drugs that any one person could be taken over their lifetime that might be impacted by their genetics? So on average, we'll start here. So these are people that only have one gene affected. So one of these genes, we're only focusing on one. So they have one variant in any of those genes. And on average, about one drug impacted over their lifetime. But when we get into people that have more and more genes affected, obviously you can see the number of drugs that might have been influenced by those genetic variants went up as well. That's what you would expect. But we even have a case, this person had 3 genes that had variants in it. They inherited 3 different variants, which affected over their entire lifetime 14 different drugs that they took. So this is important information that, again, the FDA recognizes that we should be all aware of, which includes my dad, who disregarded my recommendations. Thankfully, he's still here. So Plavix. So anyone on Plavix. But if you're already on the drug and you're stable on the drug, then there would be I don't want to give any clinical recommendations here, but just be aware that genetics is important for drug prescribing. And so the next thing we want to do, and this is really the important thing, is what's the impact when it comes to adverse drug events? Just because we're carrying the variants doesn't mean you're going to get an adverse drug event. Just because you have the drug doesn't mean you're going to get an adverse drug event. There's a lot of factors that contribute to your risks. Drug-drug interactions, age, what you ate. So some of the drugs, if you ate a salad, is not good for you. But of course, genetics is important as well. And what I'm graphing out here is over 300 adverse drug events that occurred in this population of 300 individuals. These are the drugs that patients had, that the adverse drug events were associated to. I talked about warfarin. We have codeine, simvastatin, clopidogrel. The clopidogrel is Plavix, so for those that might be aware. And they're just grouped out by specialty. But what we found when we looked at all the variants and all the drug information is that people that carried the variants and were given the drug typically had about a 2-fold increased risk of having an adverse drug event than those people that didn't have the variants. They were the normal, non-mutant form. Again, emphasizing that there's potential clinical implications when we're giving these drugs based on our genetics. The next question is, well, when would be the optimal time to get genetic testing? Well, our genetics doesn't change when we're born. So we could all get genetic testing when we're first born, but how do you maintain that information? How does that information get transferred from when you move from Wisconsin to Minnesota to Michigan or anywhere else? So maybe a healthcare institution would curious to know, well, at what age will you get the most benefit? So I showed this graph before. About 80% of the population carried a variant, carried the drug. That's our starting point. These are called survival curves. This is a death curve. So at age 20, all our 300 people were alive. At age 90, about 20% are still alive, 80% are dead. Now this is really related to the drugs. And I have two different perspectives. One perspective is based on the 300. Like we have a numerator and a denominator. The bottom part is your reference. In this case, the percent of the 300, your denominator is always going to be 300. And the numerator is going to be, well, what fraction of the population at that age could have benefited from pharmacogenetic testing before they were given the drug. Whereas the living, your numerator is consistent with this, but your denominator can change based on who's living at that age. I hope that makes sense, because as the population ages, some of the people are going to die, but there's still people that are alive that may still benefit from pharmacogenetic testing. And so we'll focus first on the dotted line here, this blue dotted line. So at age 50, about 80% of the population could still benefit from pharmacogenetic testing, meaning that you don't have to test someone when they're born. You can wait till they're 50 and you still be able to use that type of information for the rest of their lives. But as you can tell, as the population gets older, patients are now being given the drug and patients are dying. And that's why this curve is going down. Conversely, when we look at those percentages that are still living, who might still benefit from pharmacogenetic testing, what you can see here is the line keeps going out before it starts heading down, meaning that the people that are still alive can still benefit from potential testing at the pharmacogenetics. So it's never too late. So I'm going to try to wrap this up, and I recognize I talked about two very different studies, one in twins, studying thousands of diseases, and then we had the pharmacogenetics. Again, it's all rooted in genetics. It's something we all inherit. And I would emphasize, as we walk out the room, that all of us are carrying variants. We're not only carrying variants that are related to how we respond to drugs, we're all carrying variants that are related to common diseases, seborrheic keratosis, breast cancer, or anything else. We're all carrying variants. That doesn't mean we're all going to get disease, but we all have these variants. And some of us, unfortunately, carry very rare variants that are results in high instances of cancer or cystic fibrosis or what we call Mendelian disease. But we're all carrying them. So it's important to recognize that we're all unique, but we're no different from one another because, again, we're all equally bad at the genetic level. And in the context of pharmacogenetics, again, the vast majority of us will, at some point in our life, will be given a drug that may be impacted by our variants. Emphasized by 80% of the population that we studied was given the variant as well as given up to 2 drugs. And I gave an example where one patient had 14 drugs that might have been impacted by their variants. Again, all this information is all inherited from our parents. And one of the greatest predictors of human health, it includes family histories, and I think this is important not only in the context of drug prescribing. So if you have a mother or father that responded poorly to a particular drug, you should be a little bit more careful, cautious on how you might take that given drug. But I want to emphasize that based on our twin data, few diseases are really rare or are random. And I believe because of the shared environment and because of the shared genetics, that all of us, again, are carrying these variants, and we just need to look to our parents to give us an inclination about what our future is. And then again, families linked to an electronic health record can be used to study human disease and understand the genetics. And that I wanted to emphasize based on what I showed with seborrheic keratosis. So with that, I'm going to end. Hopefully there'll be enough time for Q&A. There's a lot of people that were involved in this study. I'd like to, again, thank my parents for sponsoring and allowing me to come, and everyone else for coming here. I'd like to thank my funding organism, or funding agencies, and here's my political plug. Please support politicians that support research, not cut research by 20%. But nonetheless, none of this work could be done without any of these people and the funding that I was able to acquire to do it. So thank you for your time, and I look forward to a lively discussion.
Mary and I are going to wander around with the microphone. If you have a question, please wave at us, and we'll hand you the microphone and give you a chance.
Early on, you talked about doing twin research, but then I was unclear whether this was just monozygotic or were you doing both?
We were treating the twins as any other sibling relationship because we don't know the zygosity in the EMR. We've actually done some studies to look at height differences and weight differences and other information with an electronic medical record to try to learn who are more likely to be the identical twins versus the fraternal twins. But unfortunately, they're not specifically characterized with an electronic medical record, unless if they have, maybe they've gone through genetic testing because one twin has a disease and they wanted to confirm that the other twin may or may not be at risk for the disease. That's the only time that we're able to extract the zygosity information. But that of the 16,000 individuals that we have, only 200 of them we can reliably extract the zygosity information out of. So we just looked at, very simply, what is the risk of the one twin being diagnosed if the other one is, with the understanding that those values are going to be influenced by genetics and environment. But we believe environment's going to play a large role in that.
A couple of years ago, I read about a software program that was written about issuing and testing on drugs in terms of, I use Xarelto, he uses Plavix. I've been very successful with that. He has not been with his Plavix. However, Joe Blow, on the other hand, was really good with the same basic genetics as him, and he did lousy on the Plavix, but very well on Xarelto. The program actually allowed for a distinction in terms of I can use this and you can use something else, and both are successful. But the problem, as I understood it, was letting that information out because of the restrictions on information on patients has it locked up. It's not moving at all. How would that affect in your situation in terms of that evaluation?
So the information that we got, all the patients volunteered all their information and their genetic information. But they're obviously dead, and so it's not going to benefit them anymore. But two things. One is we promised them when they signed up for research that we wouldn't share their information. They actually preferred us not to share that information. And I understand hesitation of sharing genetic information because of possible discrimination, worried that insurance companies might gain this information, et cetera, which is illegal, but nonetheless. The second thing is none of our experiments were done in a clinical setting. So by law, we're actually not allowed to release any of that information to individuals, even the people that are alive, because there's a lot of regulations that dictate on how a test can be run. And in research, we don't follow those regulations. We just need to get the results, and there's just a lower bar And because of those two reasons, we can't share that information directly back to the patient. Did that answer your question? And likewise, we can't share it to their physician as well. We're doing other studies, though, where we actually...
So how do we utilize that valuable information? We as a public, the general public, if we don't do DNA testing at some point in our lives, then we're at shaking the dice in essence.
To some degree, yes. There's useful information in our genetics that we're not using, and we could dedicate a whole nother hour on why that is the case. Insurance companies don't want to pay it. They don't want to pay for a patient that may not need it. So many of us, I mentioned 3 to 4% of us are walking around with known Mendelian conditions. That might be a little overestimated in this population, because I recognize that maybe your younger folks that may have inherited those things are no longer in this room, given the demographics. But nonetheless, an insurance company doesn't want to pay $1,000 for a test that only benefits a small percent, and there's no guarantee that the person will actually get it. So preemptive genetic testing doesn't work. If you get breast cancer, they're more likely to do the test. Unfortunately, we don't do preemptive because, again, there's no appreciation, especially with insurance companies, on how to use that information to prevent disease. And then doctors, and again, I'm overgeneralizing here, but physicians don't really know how to interpret a lot of this information as well. So they're already bogged down with everything and all this charting. They hate electronic health records because it just adds more time and more documentation. And to add on more information like genetics, they don't know what to do with. So there's resistance to get this type of information readily accessible and used.
Is there any question or a connection between the variants and the origins of your ancestors, say Asian or Northern European ancestry?
Yes. So if you looked at the genome of someone of European ancestry compared to the genome of a Native American, or the genome of a Russian, or the genome of a Chinese person, they're going to have very different variants. And they might have variants, but the frequencies in that they might have same variants, but the frequencies might be different in the population. So maybe 30% of us carry a variant. We're all presumably more or less European. 30% of us at a very specific position carry a variation. But if in an ultra universe and someone was giving this talk in China, maybe that frequency is only 5% of the population carry that variant. But so there's differences both in the variants that we inherit, but there's also differences in the variants that, in the frequency of the variants that are shared. Does that answer your question?
I'm an identical twin, and several years ago, I had an ovarian cyst removed. A couple years later, my twin sister also had an ovarian cyst removed. The results that came back showed that each time there was another individual being formed. My sister and I were number 13 and 14 in our family.
Wow.
Could have made 15 and 16.
Question
I don't know if I heard I heard it was an observation, but what's the question specifically?
No, it was just an observation about identical twins and having the same thing happen.
Well, I'm very glad that you're still with us, and I hope you're a twin sister as well.
She is?
Good. Yeah. And I don't know if you had any genetic testing or not within the family. There's no guarantee that the cause of this is due to genetics. And even if it is, we don't know everything about genetics. Typically, ovarian cancer and breast cancer can go hand in hand. And those are the BRCA genes. Those are commonly known.
Do twins run in families?
Do twins run in families?
I've heard that, but I don't. Twinning is more common in African American population or African populations than Caucasian populations. Now, whether or not that is actually based on the difference in genetics between the populations or the different environments, I'm not sure. But I've heard that they run in families. It's just what's influencing that, I don't know. It could also be it could be environmental. So what are the chemicals in the water for that particular family that lives in one area? And it's true. I mean, we're all consuming stuff we may not recognize, but there's a lot of stuff that we're being exposed to. And everyone's a little bit different based on where you live and where your family is and what you ate and everything else.
Mary's side of the room is really beating us out on good questions. Thank you for good questions.
When might this information be available to clinicians who will be willing to accept it and look at it and have time? When do you see that?
I envision that this is going to happen very soon. So I talked about our biobank. We have 20,000 patients. These are all patients that are linked to electronic health record. We're one of a few institutions in the United States that have similar biobanks. Some of them are larger, like Mayo Clinic has about 50,000 patients all linked to electronic health record. They don't have as good data as us because many of their patients are referrals, and whereas ours is a very stable population. Vanderbilt University, Northwestern, all these institutions have these biobanks, and we all work together to advance this field. But because of this, the federal government has actually initiated what's called the All of Us program, where they're going to recruit 1 million patients into one central biobank to allow researchers to study. All these patients are going to be linked to electronic health record data with genetic information, and all these individuals are going to volunteer their information. They're going to be specifically recruited. And we're going to be able to look at not just 300 people, we're going to look at 1 million people. And we're going to do this in a clinical setting. So all these people that are going to be donating their DNA will also be, all that genetic information that will be acquired will be done in a clinical manner. So it's hybridizing the genetics with the research, or sorry, the research with the clinical practice. And so this is research on clinical practice. And because we're doing those studies and we're involved with that, we're going to be able to start initiating and pushing the field forward by doing these really large studies demonstrating there's a lot of important information within our genetics that we should be utilizing in the electronic medical record. Now, there's still challenges, is like, how do you report that information? What variants do you report? So someone is risk for Alzheimer's disease. Do they want to know that information? And okay, well, maybe they do, or maybe they don't. But even if they do have that information, can't do anything about it, unfortunately. Not a whole lot. There's a lot of diseases like that. So there's still a lot of debate in the field, and what information can we acquire, what is going to be most useful, and what is ethically put on us, what is ethically, what should we disseminate? Because sometimes patients will say, I want to know everything, until they know everything.
We have time for a couple more questions. Anybody? One back here, then.
I got one. Not exactly a question, but a comment and an urging to anyone who is asked, as I was at Mayo Clinic, because of a particular diagnosis, participate in a family study, participate in the research that you're doing to determine whether there is a possibility of preventing illness.
Yeah, and I can further on that, is there are laws that protect our genetics information. It's called GINA. I don't remember what it stands for. Obviously, G stands for genetics, and it's about discrimination. And so insurance companies are not allowed to use their genetic information to increase your premiums or anything like that. Genetic information is not allowed, or employers are not allowed to use that genetic information to decide who to hire, who to fire. Now there's exceptions. Life insurance policies, life insurance companies can discriminate you. Actually, small companies can discriminate based on genetics. But in general, your genetic information is protected both at the federal level as well as the state level. So I would encourage anyone and everyone that is interested in getting involved, search out projects, especially if you're passionate for something. Marshfield Clinic, if you're a patient of Marshfield Clinic, we're actually going to be starting to recruit patients much like the 1 million patients I described, which I hope we're going to be part of to contribute, but we're going to have our own, where we're going to collect thousands of patients and start doing pharmacogenetic testing on all of them for the purpose of disseminating from the to the clinician and the patient to help them to determine what drugs to treat them with in the future.
Heather, when we talked about the genetic connection between disease states, when you have heart disease or something like that running in the family, does that same possibility exist with how someone reacts to a particular medication or family of medications, statins or something like that?
It's not common that a disease-causing variant influences how you respond.
Would there be a different variant though that would
Yes.
Yes.
Yes, absolutely. Now, drug companies right now are actually developing, at least this is what they say in the media, because they want to tell the population we're doing something. But there's a shift in using the genetic information that large studies of breast cancer, large studies of rheumatoid arthritis or diabetes or whatever, we're identifying hundreds of genes that are related to disease. And so once we know the biology, so pharmaceutical companies are taking that biology based on the genetics and actually targeting those specific genes to treat the disease. So they're developing drugs specifically for those genes, which pharmaceutical companies aren't necessarily enthusiastic about that approach because people that carry the variants, and some of these are common, they know what happens. But what happens when the person has the variant, and then you have a bunch of people that don't have the variant, but they both have the disease? Maybe the drug only works in one group versus the other. And drug companies want a drug that works for everyone and for everything. So there's still some hesitation in fully adopting this, but they're moving in that direction.
Mary's half gets the last question.
Go ahead.
Okay, I am in a study right now for Marshfield Clinic. What are these results going to do for your program? I don't live up there. I'm not one of them.
So you are in a study or you're not in a study?
I am in a study. I just started after about 9 months of paperwork going back.
So I assume that was the consenting process?
Yes.
Okay.
And it's for breast cancer.
Okay. Now, for my research, it may not. Other than what potentially is it, in the context of what I described today, maybe you're going to be in our system. So I don't know you're in the system. I don't know who you are in the system. But based on what you're telling me, you're likely being treated in a Marshfield Clinic system hospital or outpatient clinic. So you're in the system. That information is being evaluated daily from an anonymous perspective. So we don't actually know who is who, but we know who has breast cancer and who doesn't. We just don't know what Johnsons have breast cancer and what Smiths have breast cancer or anything else. Now, in the context of my research specifically, if you have family members, and if just based on the longitudinal data that you happen to live together with a family member and everything else, then maybe you might also be in the context of the large family study that I described before.
I'm going to give Joan Wasserberg the last word.
You pronounced it correctly, right?
I did. I think I got it fine.
We have a grandson who was adopted at birth, and there are no relatives. And all through his growing up time and his now as an adult, many, many weird things, maladies. And I've often wondered about his genetic background, but we never know it. Would he benefit at all from genetic testing?
I don't know if I can answer that. I think everyone can benefit, but to what degree is the big question. I mean, we can all benefit to understand a little bit about our history. And a gentleman was talking to me earlier saying, did Ancestry.com or you do 23andMe or whatever these genetic tests that are more recreational, and they're going to tell you you have X% from Great Britain versus German versus France versus Neanderthal DNA. So knowing that can benefit someone. It may not clinically benefit, but maybe there's something there. But maybe there isn't. So I can't say with one person how much benefit. Obviously, from pharmacogenomics perspective, everyone's going to benefit. It's just whether or not that information is actually going to be used, which means that it's not going to be benefited.
But you should have a basis.
Well, I would like to thank.
I'm sorry. Say again?
It's easier and or better if you have a basis of comparison.
Yes, I mean, understanding also the histories of your parents is going to significantly benefit. So if you have a genetic variant for a blood clotting disorder and no one really knows what that variant is, but you see a parent also with that variant and has the same condition, then you can start making connections that that variant is actually important for the disease. So having family information is really helpful when trying to understand people's diseases.
Well, I'll try again. I wanted to thank John and Karen Hebbring for their sponsorship. I thank all of you for coming. This is a terrific turnout. And especially to thank Scott for a fantastic presentation.